Hello students, what we will be seeing in this first video is the history and development of medical microbiology. That is the historical events that led to the development of medical microbiology as a specific subject. In fact, when I say that the history of medical microbiology, what I am actually mentioning is the history of microbiology as a science. Because it is the medical microbiology, it is the microbiological developments in the medical field that led to the proper formation of microbiology as a science. So most of the events that you will come across will you will already be knowing, because this you must have already studied in your history in the first paper. Okay. So what you will be seeing under this particular topic is, first is the definition of medical microbiology. Then you will see who are the early contributors to this field, the pioneers of medical microbiology, and then a specific subject came out from this medical microbiology, that is virology, after the discovery of viruses and so many other inventions in this particular area. And then after this comes the science of immunology as a specific subject again, because the immunology and medical microbiology are interrelated subjects. So a specific science of immunology came out from this particular medical microbiology field. And then you will be seeing the developments made in the treatment of various diseases, that is chemotherapy, and what are the various recent advances that have taken place in this particular field, that also you will be seeing. And finally, I will mention some of the Nobel laureates who actually worked in medical microbiology and got this honor. Medical microbiology can be defined as the study of causative agents of infectious diseases in humans, their pathogenicity, methods of diagnosis, control measures, and methods of treatment against these diseases. So, in simple terms, what we can say is, in medical microbiology, we are going to study about the microorganism that is responsible for a particular disease in human beings. So study means what? What are we going to study about that organism? We are going to study its pathogenicity. That means how this organism enters inside the body, where does it go inside the body, how does it multiply inside, what are the various substances it releases when it grows inside the body, and what are the symptoms you are going to observe in the body when this organism is multiplying. All these will be dealt with under pathogenicity. Next is, how are we going to detect this disease in the affected person, in the patient. So that will be the next study. And then how are we going to control the spread of this infection in a community? That is control measures. And we are also going to see how are we going to treat the person from this disease. What are the various antibiotics that could be given to this person to cure him from the disease. So all this will be covered under medical microbiology. See, in ancient times, what people were thinking was, whenever they see any disease in a community, in a few population, they used to think that this disease is because of some supernatural power, either Devan or Devero. They don't know. They thought that it is because of the anger of some supernatural powers, or they thought that it is because of some dirty environmental factors like dirty air or dirty water or dirty food. So diseases may be because of that, or they thought that diseases may be because of some bodily constitution. ಕೇಳಿರ್ತೀರ ನೀವು ವಾತ ಪಿತ್ತ ಕಫ ಇದರಲ್ಲಿ ಏನಾದರೂ ಇಂಬ್ಯಾಲೆನ್ಸ್ ಆದರೆ ಅದರಿಂದ ರೋಗಗಳು ಬರುತ್ತೆ. So they used to think like that, or the diseases might be due to some faulty diet, some improper diet. Because of that, diseases may come. So this is how ancient people used to think. ನೀವು ನೋಡಿದ್ರೆ ಇದರಲ್ಲಿ ಕೆಲವೊಂದು ಸತ್ಯ ಹೌದು. ಈಗ ಬಾಡಿಲಿ ಕಾನ್ಸ್ಟಿಟ್ಯೂಷನ್ ಇಂಬ್ಯಾಲೆನ್ಸ್ ಇಂದ ಡಿಸೀಸ್ ಬರುತ್ತೆ ಅನ್ನೋದು ಕರೆಕ್ಟ್, ಫಾಲ್ಟಿ ಡಯಟ್ ಇಂದ ಡಿಸೀಸ್ ಬರುತ್ತೆ ಅನ್ನೋದು ಕರೆಕ್ಟ್. ಬಟ್ ಕೆಲವೊಂದು ವಿಷಯಗಳು it was not proved at that time. ಎಲ್ಲಾ ಡಿಸೀಸು ಇದರಿಂದನೇ ಬರುತ್ತಾ? ಇಲ್ಲ. ನಮಗೆ we know now that there are diseases which are caused by faulty diet or bodily constitution, and there are diseases which are caused by some infectious agent. So this idea, this knowledge was not there during ancient times. So the concept that diseases might be caused by some invisible organism in the environment was put forward first by an Italian entomologist, that is Agostino Bassi. Agostino Bassi showed that a particular disease in silkworm was caused by a fungus. In fact, in his honor, the fungus was named as Bavaria bassiana later on. He didn't name it like that. It was later named as Bavaria bassiana. So this was the first disease to be shown caused by an invisible organism, a small microorganism. We all know this person, right? A very popular face in microbiology. Yes, Louis Pasteur. Louis Pasteur was a French chemist basically, but while he was doing his work on fermentation, he got interested in microorganisms, and thus he started studying about the nature of microorganisms. While continuing his studies, he proved the germ theory of disease. That means diseases were caused by microorganisms was proved by him, and he also is responsible for the control of many dreadful diseases of those times. I have mentioned a few here. That is pebrine disease that was affecting the silkworm, and he controlled the pebrine disease in silkworm by a very innovative method. And then he also developed immunization programs for anthrax disease that was afflicting cattle, and chicken cholera disease that was affecting chicken industry. And he also successfully developed a vaccine for rabies disease and tested in human beings. During the 19th and 20th century, surgeries were already in practice. Many doctors were doing operations or surgeries, but the only problem was that many of these surgeries, though they were successful, the patients used to die after the surgery because of some problem, especially because of infection in the wounds caused during or after the surgery. So doctors couldn't understand why this was happening. Now, after reading the work of Louis Pasteur, Joseph Lister, an English surgeon, got this idea that it is maybe because of the germs or microorganisms that is present in the air, in surgeon's hands, or in the instruments that is used. Maybe because of these germs, the wounds are getting infected and deaths are happening. So with this knowledge, what he started doing was he started using an sanitizer, or he started using a chemical which can act as a sanitizer, that is phenol or carbolic acid. Now, while doing the surgery, while the operation was going on, he used to spray this phenol or carbolic acid in the environment where the surgery was happening. He used to swab the instruments and also the beds and also the clothing, everything with phenol or carbolic acid, which acts as an antiseptic or a sanitizer. So after doing this, the death rate drastically came down after the surgery. So with this success, he could very efficiently control the microbial infections in patients. And that is why he was called the father of antiseptic surgery. So slowly the concept of germ theory of disease started gaining hold after the work of Louis Pasteur and also Joseph Lister. Who is this serious looking person? Yes, it is Robert Koch, who was a German physician. While Louis Pasteur was working in France, he was doing his studies in Germany, and most of his work has been very efficiently utilized by Louis Pasteur to develop his vaccines. In fact, if you could see that most of the bacillus that is responsible for diseases was discovered by the Robert Koch laboratory. He discovered the bacteria that is responsible for cholera, and the bacteria that is responsible for tuberculosis, and also the bacteria that is responsible for anthrax, Bacillus anthracis. So most of the basic work was done by him. In fact, we all know that his laboratory developed most of the pure culture techniques that has been in use even today. He was the one who developed most of the staining techniques. He formulated a criteria to prove that a specific organism is responsible for a specific disease. I repeat, in his laboratory, they came out with a protocol to prove that a specific pathogen or a specific organism is responsible for a particular disease. And this we call as Koch postulate. What is it? We will see in the next slide. In Robert Koch's period, the concept of germ theory of disease, that means a microorganism is responsible for a disease, has already been accepted. So what happened was, there are laboratories from different parts of the world who started claiming that a particular organism is responsible for a particular disease. There were too many confusions. Different laboratories came out with different organisms saying that this is responsible for a particular disease. So to clear all these confusions, Koch gave out a set of rules, a set of protocols to claim that a particular organism is responsible for a particular disease. And this has been put forward as Koch postulate. So Koch postulate consists of 4 steps. The first step here is you have to isolate a particular organism from a diseased animal that is that has suffered from the disease and has died. So from this dead animal which has suffered from the disease, you should be able to isolate a particular organism and see to it as you also take sample from a healthy animal and observe that sample under the microscope, and you should not be able to see that organism in this sample. Only in the sample that has been taken from the diseased animal you should be able to see the organism, and from a healthy sample you should not be seeing that particular organism. This is the first thing you should prove. Once you do this, in the next step what you should do is, you have seen that organism in the sample, right? Now, to do other 3 steps, you should cultivate this organism in a laboratory media. You should be able to isolate it in pure form. That is, only that organism should be there. No other culture, no other organism should be there in your plate. So, you should be able to get it in pure culture. This is the second criteria you should satisfy. Next, what you must do is, take the pure culture of this organism and inoculate it in a healthy animal, the animal which does not have any disease. Inoculate that pure culture in the healthy animal and observe. Now, if this animal again suffers from the same disease that this animal has suffered with the same type of symptoms, then that is the third criteria that you have satisfied. You understand? Inject the pure culture in a healthy animal, leave it for some time, observe it for some time. If the healthy animal dies of the same disease with the same set of symptoms, then that is the third criteria that has been satisfied. And the last step that you need to do is, from this diseased animal, again you take the sample, and when you put it in a culture plate, if you get the same type of culture that you got it from the first sample, then that is the last criteria that you have satisfied. If you satisfy all these four criteria, then you can very clearly claim that this particular organism is responsible for this particular disease. So this is what Koch postulate says. Once Koch discovered the causative agents of many disease, other scientists also started applying the Koch postulate and discovered the causative agents of many other diseases in the same century, that is in the 19th century. So you can see these names here, the scientist name who discovered the organism responsible for these diseases. You can easily understand the diseases here. The first one is leprosy disease caused by Mycobacterium leprae, and the second one is anthrax disease, gonorrheal disease, staphylococcus intoxication, diphtheria, pneumonia, meningitis, brucellosis, tetanus disease, and of course plague disease. So all these diseases were studied by different scientists, and they discovered the organisms responsible for these diseases in different periods of time. By the beginning of the 20th century, many diseases have been proved to be caused by bacteria. However, causative agents of diseases like rabies, smallpox, common cold could not be identified because they were too small to be seen even under the microscope. So the existence of such ultra-microscopic microbes was proved by the work of some of the early scientists like Ivanovsky, who was working on tobacco mosaic disease, and he said that this disease was caused by an organism, a pathogen, which is much smaller than bacteria. And Beijering coined the term virus for these ultramicroscopic particles or microbes. Slowly, the development of virology started gaining pace. Many people started working on viruses causing different types of diseases. And one such disease was yellow fever, which was afflicting many parts of the world. Walter Reed, he was working in Cuba, and he proved that the yellow fever disease was caused by a virus, and not only that, this virus was transmitted through the bite of infected mosquitoes. This concept of transmission, insect transmission or vector-borne transmission, was given by Walter Reed. Likewise, many other viral-oriented studies were carried out during that period. Landsteiner and Popper discovered that the viruses are the causative agent of one of the very common disease, that was poliomyelitis at that time. And in fact, during that period, Ruska developed a very complex microscope, that is electron microscope, and with this microscope people were able to see the viruses under the microscope and do a lot of developments in that particular area. And Ellerman and Bang first showed the possibility of a virus infection that could lead to a type of cancer. Generally we call it as malignancy. So viral infections can lead to cancer. This concept was given by Ellerman and Bang. And as we all know, HIV was discovered in 1981 by two separate research groups headed by the scientist Robert Gallo and Luque Montagnier. Now, I thought you might be interested in seeing this. Can you guess what these yellow dots are present here? This was one of the first electron micrograph of HIV-1 virus. This has been published in Nature, and this was one of the first photos of HIV-1. When bacteriology and virology studies were getting established among the scientists, another science also started gaining importance, and that is science of immunology. This science grew from the common knowledge that people who suffered from an infectious disease became resistant to those infections again. So this science, the history of this science, you will be studying in detail in your first unit. but I will just quickly go through the events one by one. I'm not going into details about this. The first very important contribution to this field was done by Edward Jenner, who developed a vaccine for smallpox disease in 1978. We all know the story. Of course, you will be again going through it in History of Immunology. After seeing the work of Edward Jenner, Louis Pasteur came out with different vaccines for different diseases, as I already told, for chicken cholera, anthrax and rabies. And the mechanism of this immunity that happens in human body was explained by Nuttall and Buchner. And he said that the bactericidal effect is primarily seen in the blood of patients. And Bering and Kitasato detected the antibodies in the serum of animals which were previously inoculated with small dose of tetanus toxin that they used. So they first detected the antibodies in the patient's serum. And another very important component that is present in our blood which is responsible for resistance is complement, and this was first defined by Bordet in 1895. And there is another important non-specific mechanism that we see in the body, that is phagocytosis, which is carried out by specialized cells called phagocytes. And this phenomenon was first observed and explained by Elie Metchnikoff. This is often asked in entrance exams, so please remember this name. Elie Metchnikoff observed the phenomenon of phagocytosis. And of course, Medawer and Burnett developed the technique of transplantation by immunosuppression and selection of compatible donors. What do I mean by this? See, at those times, they were doing transfusions and transplantations. You know already what are they? So when they were doing that, sometimes they were successful in doing the transplantation, whereas sometimes or most of the times there was failure. Graft rejections were happening, serious allergic reactions were happening after transplantation. So why this was happening was first described by Medawer and Burnett, and they showed that by means of immunosuppression, that is suppressing the immune system, and also by the selection of compatible donors, proper donors, suitable donors, we can effectively avoid these graft rejections or hypersensitivity reactions in a patient. Now, the concept of using chemicals to treat patients suffering from a disease came into existence, and that is called chemotherapy. And this concept was first given by Domack, who introduced a drug called Prontosil. This was in fact a chemical, a pure chemical, an azo dye. This has antibacterial effects against mainly gram-positive cocci. And even before that, Alexander Fleming accidentally discovered the antibiotic penicillin. This was a drug which has been taken from one microorganism and that can act against another microorganism. Penicillin has been taken from a fungi to act against bacteria, right? So though the drug was discovered, it was used only during the Second World War, not before that. Whereas Prontosil, people started using it even before that. And penicillin went for mass production by the efforts of Florey and Chain, and together they were awarded Nobel Prize for their significant work in this field. And not only that, after these two important efforts in this field, many other people started extracting chemicals from different sources and started using them against different types of organisms or pathogens. So the concept of chemotherapy was very much in vogue during the middle of the 20th century. But when we are advancing, do you think the microorganisms are going to keep quiet? No. They also started evolving. They also started tackling the problems that they face, and that is by means of drug-resistant microorganisms. Now there are just two slides where I'll be quickly going through the recent developments that has been happening in medical microbiology. So here I will be concentrating on mainly two things. One is how to detect the disease in an effective and quick manner. Second is how to treat the disease using different therapeutic agents. So in the diagnostic procedures, we have made two important inroads. One is by using specific molecules called antigens and antibodies that is present in the pathogens and in human body. Second is using the DNA or RNA for detecting the disease. So the new diagnostic methods rely on two things. As I said, one is on immune particles, either antibodies or antigens. Second one is on molecular substances, that is DNA or RNA. So under immunoassays, we have so many examples for detecting the diseases, so many tests that are carried out. We already know ELISA is done for detecting the disease HIV, and latex agglutination tests, co-agglutination tests are all available for detecting different types of diseases. You will be studying about all these diseases in detail in immunology. Next is using DNA or RNA, that is molecular biological methods are also very commonly used nowadays for again for accuracy and also for quick identification. So under that we have a few examples. We already have listened to a lot of this particular test, that is polymerase chain reaction. You must be hearing about this again and again every day, that is RT-PCR, which is nothing but reverse transcriptase polymerase chain reaction. And at the same way and in the same manner, we also have nucleic acid hybridization probes available to detect different types of diseases. So because of the emergence of drug resistant microorganisms or antibiotic resistant microorganisms, people have started looking for other sources, alternative sources for antimicrobial agents. I will just mention four important sources here. One is we can take antimicrobial substances from plants and we can use it as chemotherapeutic agents. So this is one area where a lot of studies are going on, and you might have heard about this particular microorganism, group of microorganism called probiotics. These are beneficial in nature. We can use them also to protect ourselves from the disease and also sometimes to cure certain diseases. And third is there are many short peptides available nowadays extracted from different plant and microorganisms called antimicrobial peptides AMPs, which can be purified and used effectively for curing the diseases. And last is lantibiotics. Lantibiotics are short peptides produced by some gram positive bacteria against others. That is, one particular group of gram positive bacteria will release these chemicals to attack other group of bacteria. So you must have heard bacteriocins, right? So antibiotic is a type of or a class of bacteriocins, and this also is being in studies nowadays, and any of these can be used nowadays, can be effectively developed as antimicrobials for therapeutic purposes. Students, the work of different scientists in the field of microbiology is so significant, is so important in managing different types of diseases that many of them have got Nobel Awards in their field in medicine and physiology. So I have just listed out these scientists. In fact, I have left some of them because of the shortage of space. And you can see there are quite a number of people here. From year to year, they have been awarded Nobel Prize mainly for their work on medical microbiology. Right from Emil von Behring's work, who said that you can take a serum from a patient who has recovered from a disease and give it to another patient to cure him. What we now call plasma therapy. You must have heard it in newspapers and seen it in newspapers. So that concept was given by Emil von Behring in 1901 itself, and he was awarded Nobel Prize for this significant knowledge. And work on malaria was done by Ronald Ross. Robert Koch, of course, got Nobel Prize for his work on tuberculosis, and many such people who worked on different types of diseases, discovery of the organism, and getting a cure for that organism, controlling that organism. So all these people were awarded Nobel Prize. People who worked on chemotherapeutic agents, who worked on viruses, who worked on immunology, and who worked on tumor immunology and tumor viruses, and a person who discovered prions, a new type of infectious agent. Stanley B. Prusiner also was awarded with his highest honor, and this is another organism, Helicobacter pylori, which was responsible for stomach cancers. So all these discoveries itself are so significant. They got this highest honor because of that. So even even in the last year, in 2020, Alter, Huckton, and Rice got Nobel Prize for the discovery of a new virus, that is hepatitis C virus. So students, that is in short about the historical events that led to the development of medical microbiology. I have thrown light on a few events here so that you can get interest and to know more about the history and also the development of medical microbiology. Now what I want you to do is just pick up a particular word from this presentation. For example, you can select antibiotics, or you can select antimicrobial peptides or probiotics, or any such word which is new to you, and do more studies on it, do more research on it, and write an assignment, and submit it when you come back to the college. Will you?